Hello, I'm Klaus Mueller from Stanford University. Today I'll present our paper, RadVolViz, an information display-inspired transfer function editor for multivariate volume visualization. It's the PhD dissertation of my student, Ayush Kumar, who is the first author of this paper. This paper is a joint collaboration between Stanford University, Brookhaven National Lab, and University of Irvine, California. And enjoy the show. So the core application areas of this research is mostly on material science for battery research, like, for example, lithium-ion batteries, solid oxide fuel cells. And these are things developed at Brookhaven National Lab and also at the University of Irvine. And common tasks really are determine the distribution of materials and chemical elements of these battery materials. And what scientists really want to see is detect and examine formation of grain boundaries as the battery is in use. So they look at the batteries before and after and see if the boundaries have changed. And also, while they be used, detect the formation of additional material phases. That's also something scientists are really keen of. And data sets are usually not big, like they're usually just 200 by 200 pixel slices and without 100 slices, so they're not that big. But they have more channels than usual, like they have we only seen data sets with 5 channels that mix together. So what's very important with these channels is that it's really one channel per chemical element. It measures basically the probability of an element to exist in a particular location of of this volume. So there's 5 channels for this. And in this case, or 3 to 4 or 5 channels. And some materials that you might encounter is cobalt, iron, gadolinium, titanium, oxide, all kinds of materials that are in there. We're going to see more of them later on. And they are acquired with these data, usually acquired with something like chemically sensitive electron tomography. Tomography is like 3D acquisition, or hard X-ray tomography, or fluorescence tomography. Just to give you an idea what these things look like, this is like a typical beamline in Brookhaven National Lab, high accelerated particles, X-ray particles impound on these specimen to create this image, right? So then when this is a closer look, is like this here. So it's a big instrument, right, with very high energy generated on synchrotrons. So basically, the state of the art really is what the scientists currently use is a package called TVCG, which is a public domain software developed by Kitware and other researchers. And Amira Aviso has a long history, but is also something that scientists often use. But basically what's sort of common with these interfaces are that they only have a 1D transfer function, right? Basically it can color only one channel at a time, which is a little bit problematic because you might have 3 or 4 or 5 channels. So it's really tedious to create images like this. Basically, it comes down to these design goals that we had before we developed the system. Number one, first of all, we want to visualize all channels simultaneously. We want to get away from the 1D channel. We want to get all these channels simultaneously. This is really what's the biggest shortcoming of the present state of the art was. Secondly, we want to make this semi-automatic, right? Something should come up immediately, right? That is already colored, okay, which is data-driven. And then basically you don't have to iterate along until you finally see an image, but it comes up already in some sense, right? It basically the cold start problem is solved that way. And then you should be able to select and filter to control the display complexity just to overcome these aggregation problems that I mentioned before, right? So we have a lot of interactive functionalities for this. And finally, the fourth design goal is that you should also enable manual brushing, because we've seen once these scientists have understood something about the volume, then they want to express their findings more concretely with a concrete labeling, a concrete colorization. And then they would usually publish those results in a journal or in a report. So these are the 4 design goals we had. And we designed a system that kind of addresses these design goals. So we'll input is basically the volume, this multivariate volume that comes in with multiple channels. And then what we do is basically we'll analyze them. We'll analyze correlations that might exist among these channels, and we analyze the clusters that might exist. And then we map it to this, what we call multivariate transfer function editor, which is basically a RadVolViz display and the attributes are on the periphery, and the data points are in the middle, and there's a color map underneath. So we'll talk about this in more detail later on, but basically that's what it is. But there could be millions of data points sometimes, then you'll just get too many data points in this display. So then we have a sampling procedure that downsamples this a little bit for presentation in this display. Okay, so once we constructed this multivariate transfer function display, we can now render, right? We can now actually interpolate those volumes, pass them through the colorization, pass them to the multivariate transfer function editor, obtain a color, and then alpha composite that and render it as a volume rendered image, right? With an RGB-A, right? We basically map these multiple channels into an RGB color, and then we add an opacity to it that can be defined by the user, and then we can create these volume rendered images, right? And all of this really basically fuses information visualization with scientific visualization, right? So we have the information visualization display there, and you have the scientific visualization on the right hand side. So how we use RadVolViz in this display? So basically, we'll have this multivariate transfer function editor, MTE, we call it, but we basically do this RadVolViz display, and we fuse it together with an HSL color space. We fuse that together, and this becomes like this MTE. And we basically then take these data points, the channels and the voxels, and we map them into display using this RadVolViz equation that I showed you before, right? And outcomes, when you have this, you can then basically pass the ray samples through this display, look up the color there, and then render the image from that. And this is like one image that's shown here. Okay, so that's how this goes. So now the visual interface looks like this, right? So we'll have this MT on the left-hand side, then where people can use different interactions. We'll tell you about them soon. And then in the middle is the volume rendered image. On the right-hand side are some control sliders. We'll come to talk about them soon. So at the top of them, we have frame rate indicator. Okay, so we usually render at 30 frames a second, so it's pretty interactive. Okay, so one thing we came across pretty fast was basically people wanted to not just see all of it at once. They wanted to basically focus on certain elements or combination of elements. Okay, so we devised this, what we call a lens, right? So basically everything in a lens is basically in, and everything outside is outside the lens. So this lens can be moved around freely, right? You can just pull it and move it around. And then, so if you don't use a lens, okay, then on the left-hand side, we'll see this lithium cobalt oxide data set fully rendered without any lens. All the points have contributed. And if you use it with the lens, then we only render only voxel inside the lens. Only the points that fall within the lens are rendered like this. Or we can revert it. We can only render the voxels that are outside the lens, right? We can also do that, right? So basically, the voxels are passed through. It's determined if the voxel falls inside the lens or outside the lens, and if so, it gets rendered or not, right? And we can like basically focus on regions that fit this lens criteria. Okay, so this helps actually a lot. There's a really nice interaction modulus that scientists really like. So, for example, if you put it in the middle, you get relatively homogeneous samples. So all the samples that sort of have everything inside, and this is the image you get, right? All the kind of equal weighting of all the elements, right? There's no preference of certain chemical elements. All of them are sort of expressed in the same magnitude, right? And here is one that is relatively high on titanium, as you can see where the titanium hangs out. This is the one where there's a lot of aluminum, right? Here's the lens at the aluminum coordinate, right? So now you see the aluminum where it is. It's in a different place. Or you can see where mixtures of aluminum and cobalt is, like you go in the middle between aluminum and cobalt in this MTE. Or you can go see, like you go only at the cobalt, you can see where the cobalt is. It's not the same place as aluminum. And here you can see the mixtures of titanium and cobalt. Okay, so it's a really interactive fluid interaction you can do. Just move around this lens and expose different materials. Okay, so we can also optimize the color assignment, right? So the location of the color nodes on MTE ring is not fixed, right? So you can rotate them if you like, right? And you can also optimize the space in terms of the channel correlation, right? This actually can resolve ambiguities in the color assignment. We really saw this really helps a lot because it can sort of bring out correlations and at the same time give more differentiation in color among the channels that are not correlated. I have an example for this here. So when you when you have an equispaced arrangement, then this is what this lithium cobalt data set looks like. And if you have a correlated one, you'll see quickly, you see the cobalt is all by itself, and the titanium and aluminum are correlated a little bit together, right? And now you can really see these aluminum speckles within this cobalt. You couldn't really see this on the left, but here you can see it much more clearly because the aluminum and the cobalt are really now separated in the color. Before, they're sort of washed out some similar colors. Now they're really different, and you can really see it nicely. So it's a really good way to really point out differences in the data set where there's no correlation. I can do opacity culling, which is really good, right? Because it allows you to sort of isolate dense structures inside volumes, volumetric structures, right? For example, you can take the opacity 0.99, or you can lower it all the way to 0.04, right? And then you can see, denser structures in the center, in the middle of it, right? So you can just go fluidly, just take a slider back and forth, and you'll see like different levels of opacity, right? And you can isolate this. Where this comes in handy, we'll see in a second. So you can also have one channel control the alpha. We found this very interesting because it kind of, so what we do basically, we mapped one of the elements, we map to the alpha channel, and all the others we map to the color. And that basically tells you sort of this one channel sort of determines the structure and the texture of the picture, and the other channels determine the color. And that lets you kind of see the structure of different elements within the context of the other elements. So here on the left, we use the cerium. Here we use the cobalt. Here we use the ferrum, the iron, gadolinium here, and here we use the average, right? Each of these pictures brings out different structures in that volume, and you can sort of still see what else is around, right? So we also, something that frequently requested was a slicer, like a clipping tool where you can actually go and clip the volume and into the inner, to expose inner structures. So, so basically chip away the exterior. So for example, this 4-channel lithium ion data set that we have here, this is from the University of Irvine. So where we see, this manganese, nickel, cobalt, and oxide. So one side is mostly greenish, and that shows an abundance of magnesium. Then we'll also have the other side here that shows desirable cobalt and magnesium, but it also shows this nitrous oxide, which is blue, which we don't want. That's what they said they don't want. They're really excited about this picture, actually, because they couldn't really see this before. But what they really also liked was you can clip now and expose inner structures that you couldn't really see very well from the outside, right? So basically here you can see this metallic cobalt on the left, which is this orangey kind of nugget here, and the right-hand side is greenish nugget, is nickel-rich magnesium, okay? And that is basically, both of them point to deficiencies in the sample synthesis process, right? These are like things you don't really want to see. These are like hardened materials you really don't want to see, like imperfections, right? So nickel-rich magnesium and the metallic cobalt. And so the typical workflow of scientists really is, because you don't want to clip, because you don't know where to clip, right? So what people usually will do, they use the opacity slider to reveal these interesting inner structures, right? So here you can see these structures from before, right? The magnesium and the cobalt. And then you can clearly see it, but there's something inside of it that you want to learn more about. So you expose this with this opacity slider, and then you use the clipping tool to actually see it, right? So this is a typical workflow we saw that scientists often use with our tool. So we found out that later on, because when you then look at the pictures that they publish in their journals, usually they don't look like the ones we have produced, but they're much more colorful, much more solid, colorful. So what they want to do, basically, they want to do manual coloring. Okay, so first they want to look at the volumetric structures with our tool, see what's going on, and then colorize it manually, right? So we created this manual brushing tool for this purpose. So to let them create images that can put in papers and reports, right? Basically, we thought this can really accelerate science, right? They can basically, from imaging to reporting, using just one tool, in just a couple of hours, right? You basically look at it, you make these pictures, and done, right? Put it in your journal, right? And this beats the current practice by weeks, right? They would use like weeks for this, and here we could do it very fast. So let me show you an example for this. So manual coloring, they would do in the MTE, right? They use this MTE for automated, semi-automated exploration, right? Create this image, and then they would use this tool here that we produced, where you can just take these regions that are interesting that they found, and assign different colors to them with this polygonal tool, right? So what this usually looks like, right? So basically, they would do, and this is really a sample from the workflow that they have used, right? We have seen this. So basically, where they had this data set here that they just ran out of the box, ferrum, cobalt, cerium, and gadolinium, right? And they found out there are like basically 3 phases, right? These different colors, right? There's orange, blue, and cyan, right? There's like 3 phases. But when they looked at it, they actually found an interesting 4th phase, which is this purple kind of color, or magenta, which they haven't really found before. They were a bit excited about this. And so they wanted to, this was a little fuzzy for them. So they used this brushing tool that I showed you and created this kind of picture here, where they really clearly show the structures that were there, right? They're giving like a solid color to it. And then this is basically what they usually have in the journals, right? And here, this face that I didn't know about, this purple face, they colored it in red to really make it stick out, right? So it makes a good picture of an image. Okay, so there cannot be a paper on scientific visualization that doesn't have a hurricane inside, and so we have our Hurricane Isabel that we used. Okay, so let's see what it looks like. So we basically just throw in this hurricane data. We only use 4 channels, which is pressure, speed, temperature, and water vapor. And out of the box, rendered this hurricane data set, a volumetric data set, with our MTE and this image we obtained, right? This image already tells a lot, even though it just came out, basically just read data and make image, right? There's no interaction at all in it and so on. So you can clearly see, like in the middle, you can see this temperature that's pretty high. There's like this greenish blue right in the middle around the eye, right? And it starts to go out, becomes a little yellowish. So you can see that the speed is starting to dominate. Speed is starting to dominate. It's getting like really fast there. And as you go further out, blue is starting to come in, which means like there's water vapor now, right? Because this high speed of the hurricane pulls up the water, and then it becomes part of the hurricane, right? And then further out is the red, which is the pressure, right? So you can really already learn a little bit about the hurricane just looking at this particular picture, right? For variables. And then you can focus in with those lenses, right, that we show here. Like, for example, the first picture up here shows you like the more around the eye, right? You can see the temperature really nicely. This one here, you can see this typical cloudy kind of image that has like a lot of water there, right? There's a lot of the outer layers here. And out here, you can see the pressure, right? You can see the speed and the pressure. Okay, so just by moving the lens around, you can focus on particular substructure of the hurricane and look at the structure more closely, right? Then you can also brush it, right, to really bring out your findings, right? And we brushed it like this, and then we generate these pictures from it. So, so basically the big picture is just this hurricane rotate a little bit to expose the inner structures. And then the top picture is where the minimum value is low, and the bottom right picture is where the minimum value opacity value is a little higher. Then you can basically see that the speed, you can see the higher speed, right, where it really is, and it's more close to the eye, right? You can really bring this out. So just by looking at these sliders. So final words, okay. So I showed you all of it. RadVolViz, I showed you all the details. You can see a lot more in the paper. Like the paper is actually pretty long. You can learn a lot about detail about the case studies, about the science behind it. So I encourage you to